His talk is literature-derived host coronavirus gene-to-gene interaction network of SARS-CoV-2. So yeah, Junguk, go ahead.
Thank you, Oliver, for the introduction. And you know, my talk is supposed to be one of the short talks, so I try to be short to keep some to have some time for the other discussion. So it's about and now we are back to the kind of coronavirus, but using the gene-gene interaction network. We don't have that much of results, so but I will give an introduction. So and this work is in collaboration with Oliver from Michigan and Arzucan Ozgur from Turkey. So as we all know, the COVID-19 is caused by SARS-CoV-2 virus in the future. All started in China, and well, as the up-to-date statistics, there are more than 30 million confirmed cases and more than 950 cases of death in the whole world. So this is a huge problem, and that's why we are having a virtual conference now. And everyone is trying to find the cures and develop vaccines for COVID-19. Unfortunately, there's no FDA-approved medications specifically developed for COVID-19. But a lot of many are trying to use existing drug, so kind of repurposing existing drug to treat COVID-19. Examples would be kind of remdesivir for Ebola virus, and chloroquine or hydroxychloroquine for malaria, and favipiravir for influenza. And remdesivir has been kind of found effective, and chloroquine, hydroxychloroquine, they may be effective, but there is some questions about safety, so that it's no longer approved by the FDA and for emergency use. And there are also many vaccines are currently being developed. The number is slightly smaller than what Anthony presented, but according to these vaccine tracker website, there are 46 candidates that are actively in active trials now, and there's only one approved, which is Sputnik 5 from Russia. But there's also a lot of concerns because that has not gone through the phase 3 clinical trial yet. And what's our research? So so many people, the whole world is trying to better understand the COVID-19 and their pathogenic mechanisms, and they publish a lot of their result in the PubMed and biomedical literatures. So and there's still we need to know a lot better about them. And our approach is use text mining on these kind of COVID-19 related biomedical literature and extract and analyze their kind of gene-gene interactions either between and or within kind of pathogens and host. For this, we relied on kind of LitCovid. It's one of the NIH resource. They're kind of in the literature hub collecting the peer-reviewed published articles on the COVID-19. I think this was as of kind of last week. There were more than 51,000 publications out there, and the numbers are rapidly increasing. There are a couple of thousands, for every week. And we use the tools called SciMiner. It's our in-house literature mining system that identifies host genes, pathogen genes, and interactions, and taxonomy, and some other ontological terms. And we've already shown that using this SciMiner tool empowered with ontology, that actually improves the performance of literature mining in terms of retrieving relevant document and exploring gene interaction network. And for this study, as I mentioned, we used the LitCovid. That was actually not the latest, but the one that we used is contained about 32,000 articles. And in terms of a dictionary, we try to identify the host genes and SARS-CoV-2 gene from the Ontology of Genes and Genomes developed by Oliver, and interaction network ontology describing the interactions between many terms in INO were also used in the study. And this is one quick example. This sentence, interleukin-4 and interferon gamma, downregulate the cell surface expression of angiotensin converting enzyme 2, ACE2, the SARS-CoV receptor. So those terms that are highlighted are either the genes of protein or the regulation or interactions, and or the taxon of the sentence. And so SciMiner identified these terms, and as a rough summary, we have a 25,000 kind of term dictionary for the genes, and 50 for kind of coronavirus gene, and about 200 for interaction terms. And from those in a LitCovid corpus, we got about 700 unique genes and 32 coronavirus gene and 85 interaction terms. And using these in identified entities, we kind of built a network, and this is it's simply like a co-occurrence-based at the sentence level, and this only involves those host gene that we identified. And whenever there's an interaction or edges, that means these two genes are mentioned at the sentence level in any in papers. What we were most interested at this stage is the interactions between, not only between the host gene, but between the host and pathogen gene. So this is a kind of simplified network. Again, those nodes are either host or pathogen gene. The host genes are in green, and pathogen genes are those in violet or and in diamond shape. And whenever there is an edge, that means we identified at least more than 5 papers mentioning these two entities together at the same sentence level. And there were a total of 226 such unique interactions. As you can see, kind of two separate clusters. The left one is mostly enriched about the viral entry. So those ACE2 and TMPRSS2 and those well-known genes are all in this side, and showing the enrichment entry into host on, and obviously in ACE2 and ACE, those are angiotensin system, renin-angiotensin system. The other side of the network was mostly enriched in the immune system inflammation and response to external stimuli. So that's what we got. So we got about 2 very different clusters of host and pathogen gene interactions. And another way we are looking at now is it's not only looking at the individual gene level, we also look at the pair of genes. So one gene and one host gene and one pathogen gene, like in this example, ACE2 and the structure and spike protein, and how these pairs are linked to in terms of interaction. So we are hoping this is using the interaction network ontology, and we are hoping to find any specific interactions that are related to specific pairs of host and pathogen genes. And we are also trying to improve the identification of such gene pair. We used the SciMiner and the dictionary-based identification, and we used the kind of support vector machine SVM-based scoring system between 2 entities, so that we can call kind of highly reliable gene pairs. We are now trying to use a different system. We are evaluating BioBERT, which is kind of pre-trained biomedical language representation model for biomedical text mining. It's developed by DIMS in lab in Korea. And this shows kind of performance of identifying those protein interaction or gene pairs from various the text mining corpus. And the BioBERT is actually, I don't have any comparison yet, but most of them has about 80% or higher kind of prediction accuracy, which is higher than in our current SVM, which is about 60 to 70. So we are trying to move to this BioBERT as our method. And this is still a lot of ongoing work, and we have to do a lot of further research. But some examples would be like we previously used only the abstract, but we are trying to incorporate use the full text, and eventually we used some ontology, but not heavily integrated. But we are hoping to integrate our result with the CDO, the coronavirus infectious disease ontology, and some other ontologies as well. And the network that we generated, I think we can expand them by adding some non-COVID-19 related data, so such general gene interaction, and that expands the network, and maybe we can identify some important spot or hub that may be critical for the pathogenesis of COVID-19, or potentially good candidate for the vaccine development or drug development. And as I mentioned, we are trying to evaluate the new interaction identification method and its performance. Okay, and this is again a collaboration between with other groups. So Arzucan Ozgur from Turkey and Oliver He. We have formed something called the Condor group. This is centrality and ontology-based study group. And I thank all the support for the work. And if you have any question, please contact us. Thank you.
Great. Hi, Junguk. I think you gave a pretty good talk.